Okay, awesome. Hi guys, I'm Hania Nizami. I'm going to be giving two lectures today. The first one is hormonal changes in pregnancy, fertilization, and implantation. Um, I have my number over here at the bottom and my email, so just make sure to send me any questions that you guys might have. I think that you guys have your exam kind of soon, so I will try to be really fast with my responses. The second thing I wanted to say is that the session is designed to be interactive. I know there's only 8 of us here right now, but I would really, really appreciate it if you guys could answer the questions that I have in the chat box, or if you want to unmute yourself, that's totally fine too. Also, if you have questions throughout the lecture, I actually prefer if you tell me like right after the slide or as it's going on, because I just want everyone to be keeping up, because at the end of the day, the session is for you and I want you to be benefiting. So yeah, usually the way that I conduct these is that I do a diagram or a drawing first, and then the next slide, I'm going to be quizzing you guys. Okay, so let's start with the introduction of the pregnancy. The main thing that I want you guys to notice is already highlighted in red over here. And so the first thing that I want you guys to know is that the chorion, whenever I mention chorion, what should be happening inside your head is you're thinking that this is the fetal side of things. Okay, so chorion should make you think of the fetus side. And if you hear the word decidua, it should make you think of the maternal side of the placenta. Okay, so I'm going to explain that like way more detail later, but I just want to get that out of the way right now. The chorion means the fetal side, and the decidua means the maternal side, and usually in reference to the placenta. Okay, so let's start with the steps of fertilization. So the first step is called insemination, right over here. And insemination literally just means when the sperm is going to enter into the vaginal canal. Okay, so that's the first step, insemination. The second step is a little bit more complicated. It's called capacitation. So basically what's happening is that as the sperm enter into the vaginal canal and as they enter inside the uterus, there's this process called capacitation. Now, capacitation, I want 4 things. There are 4 things that I really, really, really need you guys to remember. It doesn't have to be in this order, but I need you to remember these 4 things. The first thing is that the first step of capacitation is that you see these little orange rectangles over here. These represent glycoproteins. So basically, once the sperm enters into the vaginal canal, they're actually coated by all of these glycoproteins. And originally, these glycoproteins sort of serve to protect the sperm. But when we're talking about fertilization, we don't need these glycoproteins anymore. In fact, these glycoproteins are going to serve as a barrier between the sperm and the egg, and we don't really want that. So the first step of capacitation is removal of these glycoproteins, okay? The second step is represented by these green circles over here, and these are cholesterol. And as you can notice, there's less cholesterol. There's cholesterol being removed. And if you remember, the function of cholesterol inside of the plasma membrane is to keep it rigid. So if I have less cholesterol, that means that the membrane is less rigid or it's more fluid. Okay, so that's a second step of capacitation, is that I'm removing some of the cholesterol, and that cholesterol is going to allow for the sperm to be a little bit more fluid. Okay, and that's going to help the sperm penetrate the egg later on. The third and fourth step both have to do with calcium. Okay, so the first step is that, or the third step is that calcium is going, the membrane potential of the sperm is going to be more permeable to calcium. Specifically, it's going to be more permeable to a calcium influx. So in other words, the sperm is changing so that it's going to allow itself to allow for a calcium influx now. And the calcium influx is really, really important for something we're going to talk about in the next slide, which is the acrosome reaction. So the main thing that I want you to take for number 3 is that the sperm changes its membrane potential to allow for a calcium influx. And this calcium influx is very, very important for the acrosome reaction that we're going to go over in the next slide. The fourth thing that also has to do with calcium is that the changes that has to do with calcium are going to allow for hyperactivization. So as you can notice with the sperm over here, the tail is more relaxed here. It's more relaxed here. But at this very last part, where it's more permeable to calcium, this is going to hyperactivate the sperm. And it's going to allow for the sperm to move its tail much, much faster. And we call that a whip-like motion. Okay, so the sperm becomes hyperactive due to the calcium changes. Okay, so that's everything that has to do with the capacitation. Those are the 4 things that you need to know regarding capacitation. After capacitation happens, the sperm is going to go over here on the isthmus, and they're going to receive signals from the oocyte or the egg, and those signals from the oocyte are going to allow the sperm to sort of follow the egg and meet it somewhere here. And so this is where fertilization is going to take place. So to me, the most high yield part of this slide would really be capacitation. Okay, so remember I told you that we're going to go over the acrosome reaction in the next slide. So this is that next slide. So again, just to recap where we are, we have the sperm, it's undergone capacitation, and now it's moved over here and it's meeting the egg. And so now we're on step 6, which is, or step 5 or step 6, which is the sperm is now actually meeting the egg. So the first step is actually not pictured over here, but I want you to imagine that there's a bunch of cumulus cells surrounding the oocyte right now. So just imagine that there's a bunch of cumulus cells surrounding the oocyte, and the sperm has to release an enzyme called hyaluronidase that breaks down the cumulus cells. So again, the first step is the sperm releases hyaluronidase, which is an enzyme, to break down the cumulus cell on the outside. Then the second step is that the sperm can actually make contact with this layer right over here, this jelly-like layer right over here, called the zona pellucida. So again, right over here, my egg, the most outer layer was the cumulus cells. We broke down the cumulus cells using hyaluronidase, and now we're at the zona pellucida, which is a jelly-like layer surrounding the oocyte. Okay, and what breaks down the zona pellucida? Well, it's the acrosome reaction that we talked about before. So basically, the acrosome is the head of the sperm, okay? And what the acrosome reaction is when the head of the sperm starts to release these enzymes over here called hydrolytic enzymes. And those hydrolytic enzymes are going to break down the zona pellucida, okay? And then when we move on over here to step 3, now the sperm is exposed to these protein receptors over here. You do need to know the name of these protein receptors. I'm going to show you them in the next slide. There's actually 2 protein receptors that you need to know, but I just want to show you where it is on the diagram. So after the zona pellucida, the sperm is going to reach this layer over here where it's exposed to protein receptors, and these protein receptors are important for the plasma membrane of the sperm and the plasma membrane of the oocyte to fuse together. So once the plasma membrane of the sperm and the plasma membrane of the oocyte fuse together, the sperm nucleus can now enter into the oocyte. Okay, and this is really important because, again, something that they haven't shown you in this diagram over here is that once the nucleus enters, once the sperm nucleus enters into the oocyte is actually going to continue meiosis 2. Okay, so it was paused at meiosis 2 up until now. After the plasma membranes fuse together, it's going to resume meiosis 2. It's going to complete its meiosis 2. Okay. Another thing that's happening right after the sperm plasma membrane fuses with the oocyte plasma membrane is that there's these granules right over here inside the oocyte called the cortical granules. Okay, you see them, they're labeled over here, the cortical granules. These cortical granules, as you can see, are going to be released into the zona pellucida, and they're going to harden the zona pellucida so that now the whole zona pellucida is hard and there's no more sperms that can enter into. And this is really important because we want to prevent this thing called polyspermy. Poly means multiple, sperm is sperm. And basically what we're trying to say is that we don't want multiple sperms entering into the oocyte, because if that happens, then the egg is not viable usually, and it's going to lead to a miscarriage. Okay, so those are the important steps. I'm going to quiz you on it in the next slide. So for this first part over here, the sperms must undergo blank in the oviduct before they fertilize the egg. What was the name of the reaction? After insemination, capacitation, perfect. Okay, and then altered sperm membrane fluidity by the removal of blank. What do we remove from the sperm to make the membrane more fluid? Good, cholesterol. Okay, the removal of blank from the sperm membrane that blocks the binding to the egg. What do we remove from the sperm? It used to coat the sperm before. Glycoprotein, good. Or glycoproteins. Okay, and then a change in the membrane potential allowing calcium entry and activation of the blank reaction. What was the name of the action? Good, Ackersome reaction. Okay, calcium entry caused sperm blank. Remember, there was something that happened to the tail. There's a specific word I used for the tail. Makes the flagella go really fast. Hyperactivity, good. Hyperactivation. And then the last one over here, we talked about the polyspermy. And then second, the egg is activated to resume blank and begin embryonic development. Where was it stuck at? Where was it paused? Before. There, yeah, meiosis 2 specifically, good. Okay, so I'm going to continue testing you guys. So the most outer layer of the oocyte, what was that called? It wasn't pictured in the diagram, but I mentioned it. No, even outside of the zona pellucida. Cumulus, good. And then the layer under the cumulus is, somebody said it before. Quickly, could you go over number 2 again quickly? I don't know which one. Second, I think number 2, you mean this one. The egg is activated to resume meiosis. Basically, the egg used to be arrested at meiosis 2. And it's not until the sperm plasma and the plasma membrane of the sperm and the oocyte fuse. Capacitation number 2. Okay. The glycoproteins one, is that what you're asking? Okay, the glycoproteins is pretty simple. It's just these orange rectangles over here. They used to coat the sperm to protect it, but we want to remove it so that the sperm can make direct contact with the oocyte. That's all. Does that make sense? Okay. Okay, awesome. Okay, yeah, so then you guys already answered the second one, zona pellucida. And then what would you call the last layer of the egg? This is the part where the oocyte and the sperm fuse together. What did I call that layer? Yeah, membrane, plasma membrane. Okay, and then what was the enzyme that digested the cumulus layer? Remember that really, really outer layer? They actually have it over here depicted by these gray circles. What was the name of the enzyme? Yeah, hyaluronic is what it's made out of. So hyaluronidase is the name of the enzyme. Anything that ends with ase is an enzyme. Okay, okay, this is pretty easy, the zona pellucida. What's the name of the reaction that when we break down the zona pellucida, what do we call that reaction? Ackersome, good. And then remember I told you that there were some proteins in between the plasma membrane and the zona pellucida. Those proteins are actually isomo-1 and Juno. And because it is highlighted, I would memorize these. So again, the proteins that you saw in the previous slide, like over here, you need to know the name of them. And it's Juno-1 and isomo. Because I can see that coming up. Yeah, Juno and isomo-1, sorry. Okay, so now I want to go in a little bit more detail about the cortical reaction. So we went over the acrosome reaction, right? We talked about how the acrosome is going to release lytic enzymes, and the enzyme's going to break down the zona pellucida. Now I want to really zoom in on the part where the sperm plasma membrane is going to fuse with the oocyte plasma membrane. Specifically, what's going to happen, and you do need to know these things that I'm going to talk about, is that once this fusion occurs, as you can see over here, it's going to activate PLC zeta. Okay, and the activation of PLC zeta is going to increase intracellular calcium. Because there's an increase in intracellular calcium, that is what's going to cause the release of the cortical granules. And the cortical granules release is going to harden the zona pellucida. And because the zona pellucida is hardened, now we're preventing polyspermy. Okay, so again, the things that you need to know is that sperm and oocyte plasma membranes fuse. 1. 2, this is going to activate PLC zeta. You need to know the name of PLC zeta specifically. 3, this is going to increase intracellular calcium. 4, the increase in intracellular calcium is going to release cortical granules. 5, the cortical granules are going to harden the zona pellucida and prevent polyspermy. So I'm going to quiz you on that in the next slide. Okay, so sperm egg fusion allows for the activation of what? What's the first thing that it activates? PLC zeta, good. And then this is going to increase intracellular calcium, perfect. And then this is going to cause, what's the name of the reaction? When the granules go to the zona pellucida, what's the name of that reaction? Release cortical granules, in other words, the cortical reaction. Perfect. Okay, and then what did I say? So the whole purpose of this is to prevent something from happening, because if that thing happens, it's going to lead to miscarriage. What is it? What am I talking about? Good, polyspermy. Okay, and then really quickly, I know I didn't touch on this because I don't think it's as high yield, but we're talking about the increase in intracellular calcium, right? That is going to trigger the cortical reaction, and they also want you to know that some of the maternal proteins are also going to be translated because of this increase in intracellular calcium. I don't think it's really high yield to know. I would focus more on the cortical reaction. Okay, so now let's move on to this diagram. Okay, so let's recap where we are. We're right over here. We're at fertilization. As you can see, this is the egg, this is the sperm. Once the egg and sperm fuse together, we have something called a zygote. So the zygote is the very, very first thing. It's basically a fertilized egg, okay? And then afterwards, at day 1, day 2, day 3, the zygote is going to rapidly start increasing inside. Specifically, it's going to start doubling. So it's going to go from 1 cell to 2 cell to 4 cells to 8 cells, okay? And then after the 8 cells, it's going to really rapidly increase in size. And we call this ball of cells right over here at around day 4, the morula, okay? So it goes zygote, morula, and then once you have fluid filled inside of the morula, fluid can also be called a blastocele. Okay, we are now going to call the structure a blastocyst. So again, the difference between a morula and a blastocyst is that the blastocyst has a little bit of fluid just filled over here. So I just want you to be aware of the order of how things are happening. We had the fertilized egg became a zygote, zygote doubled for the next 3 days. After that, it rapidly increased in size to form a morula. After a morula, it formed a blastocyst. And the difference between a morula and a blastocyst is the presence of fluid. And you might call the fluid inside of here a blastocele, okay? Okay, so now we have the blastocele, right? Now we're going to zoom in and we're going to look at when the blastocele is going to implant. Really quickly, I want to touch on why there's sort of a delay. Like, why doesn't the zygote just directly go into the uterus? Basically, the reason that the development happens over here in the fallopian tube is that the uterus needs a little bit of time to prepare for the implantation. In other words, the endometrial lining needs to mature a little bit before it's ready for blastocyst implantation. And in the event that the endometrial lining isn't ready yet, or it's not mature enough, the blastocyst will not implant, and this is going to lead to a very, very, very early miscarriage. Okay, so the purpose of all of this development happening in the fallopian tube and not in the uterus is just so that the endometrial lining can develop a little bit. Okay, so now we're at the part where the blastocyst is now ready to implant onto the endometrium. So this is my blastocyst, and over here is the maternal side of things. So you can imagine this is the endometrium. This part right over here is basically this part right over here, okay? And this is the blastocyst. Basically, what I want you to know is that in the very beginning, I have this inner part of cells right over here called the embryoblast. And from the name, you can kind of imagine that this embryoblast is going to eventually form the fetus. So that's the role of this embryoblast right over here. This outer layer is called the trophoblast, okay? And what I want you to notice about the trophoblast is that it's actually going to divide into 2 parts. It's going to divide into the inner layer, which is the cytotrophoblast, and it's going to divide into this outer layer that they showed over here called the syncytial trophoblast, okay? So again, I have my embryoblast in the middle over here. I have my trophoblast, which is going to divide into two. The inner part is the cytotrophoblast, and the outer part is the syncytiotrophoblast. I want you to keep a special place in your brain for syncytiotrophoblast, because this is really, really important, because later on, it's going to start releasing hormones, actually. So I just want you to keep an eye on where it is, okay? So after the development of the embryoblast and the division of the cytotrophoblast and the syncytiotrophoblast, now the blastocyst is going to go inside of the endometrium. In other words, it's going to implant inside of the endometrium. And you can appreciate that there is now an amniotic cavity inside of the embryoblast. There's also this yolk sac right on the outside over here. And you can really see the cytotrophoblast over here. And you can see it's surrounded by this layer of cells over here called the syncytiotrophoblast. So syncytiotrophoblast is the most outermost one. So that's what's happening on the fetal side of things, okay? Now let's go on and let's switch over to the maternal side of things, okay? So again, this is the maternal, this is my endometrium. You can see my endometrial vessels over here. Basically what's gonna happen is that your endometrial or your endometrium is going to undergo a process called decidualization. Basically, the cells over here that used to be fibroblast cells are now going to mature a little bit, and they're going to be filled with glycogen. And that process is called decidualization. Okay, so the endometrial now becomes decidualized to prepare for the implantation. The second thing that I really want you to notice over here is that take a look at the endometrial blood vessels, because this is going to become very, very important. Basically, and it's not super clear here, but I'll show you later, is that part of these endometrial blood vessels are going to be degraded, and they're all going to drain into this pool right over here that's called the lacuna. So the most important thing that I want you to get in your brain right now is that a lacuna is basically a pool of maternal blood, okay? It's basically when this endometrial blood vessel is going to be broken down a little bit, create a little bit of a leak, so that I can drain over here into a lacuna. Now, I want you to know lacuna is specifically maternal. It's not fetal blood, it's maternal blood, because that's really important. And as you can appreciate, the lacuna is sort of connected to the fetus. So you can imagine that the lacuna is going to play an important role later on when we talk about the communication between the maternal blood and the fetal blood. All I want you to know right now, in case everything I said just went over your head, is that a lacuna is a pool of maternal blood. Okay. Okay, I think I didn't mention this, but I specifically want you to know that the maturation of the endometrium happens in response to progesterone. Okay, so the progesterone is what's really important. And as I'm talking about this, I actually want to talk about something. So remember how I told you that the syncytiotrophoblast over here that's surrounding the embryo is going to release hormones. It's going to release multiple hormones, but there's one that I want to focus on right now. The syncytiotrophoblast, these cells over here, are going to release a hormone called HCG. You might have heard of this because this is actually a hormone that we use to check if somebody's pregnant to begin with. So the syncytiotrophoblast is going to release something called HCG. This HCG is going to go to the corpus luteum. I don't know if you remember the corpus luteum, but basically the corpus luteum is when after a follicle gives up its oocyte, the follicle is going to kind of degenerate a little bit, and it's going to become this corpus luteum. And the normal role of the corpus luteum inside the normal menstrual cycle, like not even talking about pregnancy, but in every menstrual cycle, is to produce progesterone. So the main thing I want you to get in your head right now is that the early weeks of pregnancy, up around week 10, the syncytiotrophoblasts are going to release a hormone called HCG. This HCG is going to go to the corpus luteum, and it's going to maintain the corpus luteum. In other words, it's going to prevent the breakdown of the corpus luteum. Why do I want to prevent the breakdown of the corpus luteum? Because my corpus luteum makes progesterone, and I need progesterone to develop the endometrial or make the endometrial glands. Basically, I need progesterone to continue the pregnancy. Okay. So what was the name of the hormone that I just talked about? It stimulates the uterine glands. Yeah, progesterone. HCG is also true, but I'm talking about the development directly affects the uterine glands, like directly affects them matured. Okay, and progesterone induces the window of receptivity in the uterine blank. What was the layer of the uterus? Yeah, good, endometrium. Okay, and what do we call the pool of maternal blood that has the maternal blood from the rich capillary beds of the postcapillary venous lakes? Lacuna, good. And it's maternal, okay? It's maternal blood. Perfect. Okay, and then the syncytiotrophoblast, what was the name of that hormone that it creates? The syncytiotrophoblast. HCG, perfect. And what's the role of HCG? It's preventing the breakdown of corpus luteum. Perfect. And the corpus luteum is going to make the progesterone. Okay, so for this one, the first two embryonic cleavages, then we have the morula. The outer cells of the blank begin transporting fluid into the embryonic mass. Does anybody know what that is? The outer cells of the morula. I think that was a bad one. But basically, just what I want you to know is that the morula is the earlier stage and the blastocyst is the later stage. That one wasn't really clear. Sorry about that. And then after morula, what do we call the next stage? Once it develops a seal or a blastocele, what's it called? Blastocyst. Perfect. And the blastocyst is what's going to implant. Okay, the next thing I want to talk about, there's two things actually that I want to talk about on this slide. Firstly, I want you to notice that this is my morula and this is my blastocyst, right? Notice that the zona pellucida is actually going to break down and go away. That's one thing I want you to notice. Morula has a zona pellucida, blastocyst does not. Second thing I want you to notice is that if you look at the cells over here, the embryoblasts, they're actually going over here on one side. And when something goes to one side, we call this eccentric, right? So this eccentric embryoblasts form an embryonic pole. So basically this part over here, this section of the trophoblast right over here, we're going to call this the embryonic pole. And this specific part right over here is where the blastocyst is going to attach to the endometrium. Okay, this part right over here, the embryonic pole. Okay, I'm just going to answer this one because I don't think I explained it. But yeah, the trophoblast, so specifically this layer, is going to secrete enzymes that's going to break down the zona pellucida. So if I show you over here, the outer layer that they didn't really show over here, the trophoblasts, that's going to break down the zona pellucida. And then remember I told you that endometrium is going to change? What do you call that change? Or the presence of those new cells that now have glycogen? The maturation of the endometrium has a specific name. Decidual. Yeah, decidualization. Good. And you can see the decidual cells over here. Okay, so what part of the blastocyst secretes HCG? Syncytiotrophoblast, good. And the role of the HCG is to keep what alive? I know I asked this before, but I want to ask again. HCG maintains the corpus luteum, and the reason to keep the corpus luteum is to increase the progesterone. Perfect. Increase progesterone. Okay, good. And this is up until about week 10, okay? We'll talk about what happens after week 10 a little bit later. Or actually, I can tell you right now. But basically, after week 10, the syncytiotrophoblasts are going to be able to make their own progesterone. Okay, so at around like week 8 to week 10, the syncytiotrophoblasts is no longer going to make HCG because it no longer needs to maintain the corpus luteum because it by itself can make the progesterone. Okay, and that happens around week 10. Okay. Now, remember when I told you about the lacuna over here, and I really, really emphasized to you guys, lacuna is a pool of maternal blood, right? Exactly, maternal blood. And the reason that I keep emphasizing that to you guys is that later on, you're going to see the development of the chorionic villi. And I just want you guys to know that right over here in the lacuna is sort of where all the maternal blood drains into. And then after the maternal blood drains into, it's going to start diffusing things to the fetus. And some of the things that it can diffuse is, for example, gases. And gases just passively diffuse across the membrane. You could also have facilitated diffusion. Facilitated diffusion means that it's passive, but you need a protein. And an example of that would be glucose. So for example, the glucose from the maternal blood over here to enter into the fetus would need a special protein for it to transfer. And we call that facilitated diffusion. Sometimes you need active transport. When I say active transport, that means I need a ATP using transport protein. And an example of that would be amino acids. And the last thing that I want you to know about is this process called transcytosis. Transcytosis basically means that a vesicle is going to leave from the maternal side and it's going to fuse onto the fetal side, and that's how it's going to go. It's basically going to go across using a vesicle. And an example of that would be iron. I don't know if you need to know these specifically, but I would at least know that like gases, so like oxygen, carbon dioxide is just completely like simple diffusion, doesn't need any proteins. I would know that glucose is facilitated, which means it needs a protein. Amino acids are active, and then iron is transcytosis. I don't know how high yield it is, but I would at least familiarize myself a little bit. Main idea, in case everything I just said went over your head and you're like, what are you saying, Hania? All I need to know is that there's a bunch of stuff going from the maternal side to the fetal side. Examples: oxygen, glucose, amino acids, iron. All of that needs to go from the mother to the baby. Okay, and then you guys already, could you please repeat what happens after week 10? Yes, after week 10, the syncytiotrophoblaст is going to stop making HCG. It's going to make its own progesterone. Is that clear? Okay, awesome. And then you guys can probably already answer that. The lacuna is filled with what kind of blood? Maternal, perfect. I want that to be super clear. Okay, so now moving on. What I want to show you over here is that we can now actually see the placenta starting to develop. Okay, and I want you to know the placenta has two parts. Remember I said this in the very beginning. There's the fetal part, and specifically what I need you to know is that look at this part over here. Look at how it has so many villi. Okay, this part with so many villi is specifically called the chorion frondosum. And it's basically the part of the placenta that comes from the fetus. You see these blue projections over here? The chorion frondosum is the fetal part of the placenta, and it's very, very, very villous. Okay, that's what I want you to know. Second thing I want you to know is that the second half of the placenta is made by the decidua basalis, and it's over here. And this is the maternal side. Fetal side, blue, maternal side, orange. Okay? Next thing that I want you to see is that you can see surrounding the fetus, I have this thing called an amnion. And inside of my amnion, I have an amniotic cavity. Basically, this is just a cavity that allows for the fetus to develop, and it has fluid inside of it so that the fetus can move around and it's not like constricted. That's the main thing that I want you to know. Okay, going on. So imagine looking at this villi. Imagine that I zoom in on this. Basically, if I zoom in on the villi, I'm going to have this diagram over here to my right. So here's what I need you to know. I need you to know the development of how the villi come, right? So first we have the primary villus. The primary villus is made of the syncytiotrophoblast on the outside and the cytotrophoblast on the inside. This is the primary villus. Secondary villus, the inside the part that used to be filled with cytotrophoblasts, is now replaced by mesenchymal cells. Mesenchymal cells are sort of like supportive cells, like fibroblasts, stuff like that. And then finally, we have the tertiary villus. And the tertiary villus has my fetal blood vessels right over here, as you can see. So just, again, imagine inside of this villus, you would have the fetal blood vessels over here. And you can see it's in very close contact with the maternal side. So you can kind of see where this is going. We're getting closer and closer to where we're going to see the transport of nutrients from the mother to the baby. Okay. So we have the primary villus over here made of syncytiotrophoblasts. What makes the secondary villus? What fills the column to make it a secondary villus? What kind of cells? Mesenchymal cells. Perfect. And then what about tertiary? The presence of what? Fetal blood vessels. Good. Okay. What did I call the part of the placenta that was from the fetus? That was really, really villus. You guys remember? It was near the embryonic pole. What did I call that? Chorion. Chorion is good. Chorion frondosum. There we go. That was the answer I was looking at. Chorion is right, but like a more specific one is chorion frondosum. Okay. What do I call the maternal side of the placenta? Situa-basalis. Good job. And what develops around the fetus so that I can move around? So that's not restricted. Amnion. Good job, guys. Good. Yeah. Amniotic sac. Okay. And then something I didn't really talk about, but I can show you over here, is that eventually, a little bit down the line, is that this amnion and this chorion, this blue part over here, are actually going to fuse together. And we're going to call that the amniochorionic membrane. It's nothing really high yield, but you just want to know that these two things are going to fuse together. So this white part over here is going to disappear because these two membranes, the amnion and the chorion, are going to fuse together. Okay, so now we're at the part where I can really, really zoom in to what's happening when we talk about maternal and fetal exchange. Like, how does the blood go from one thing to another, right? So let's look over here. I have the maternal blood, and they're surrounding the chorionic villi. So again, let me just try to orient you with the previous thing. Basically, what I'm looking at over here is I really zoomed in on one of these villi, and imagine that around the villi, I have a bunch of maternal blood, okay, surrounding the villi. So I have a bunch of blood. Like, imagine I took a cross-section of the villi, basically. Like, I just cut it cross-sectionally. So you can see I have the outer layer of the syncytiotrophoblasts, the inner layer of the cytotrophoblasts, and then inside I have the embryonic blood vessel. And again, this should make sense to you because we just went over the steps of the chorionic villi. We talked about there's outer layer of syncytiotrophoblasts, inner cytotrophoblasts, and we talked about the tertiary villi are going to have the fetal blood vessels. So that's what you're seeing over here. This is the fetal blood vessels. Something that they want you to know is that some parts of the villi, they actually don't have the cytotrophoblasts. You see these parts over here? They don't have the cytotrophoblasts. And the reason that this is that it actually makes it easier for any of the things from the mother side to enter into the fetal circulation. So there's some parts that are very, very thin, so that anything from the maternal circulation that's represented right over here can really easily diffuse across. Okay, so basically, the main thing that I want you to see is that there are 3 layers composing of the chorionic villi, or there's 3 things that the maternal blood needs to pass through to finally get to the fetal blood. And you do need to know them. First one is the syncytiotrophoblast. It's the most outer one. The second one is the basement membrane, shown over here. And the last one is just the endothelial cells surrounding the embryonic or the fetal blood vessels. Okay, so those are the 3 layers. You have the syncytiotrophoblast, basement membrane, and the endothelial cells of the fetal blood vessels. Okay, so what do you call the fetal side of the placenta? Specifically the projections. What do I call those projections? Chorionic villi, perfect. And they're lined by what? What's the outermost lining of them? Syncytiotrophoblasts. And the in the middle at their core, what do they have? Inside the tertiary villus. Yeah, endothelial cells and fetal blood vessels, basically, yeah. Good. Okay, I told you that some parts of the villi, they lack some cells. What kind of cells do they lack? Cytotrophoblasts, good. Okay, so now let's go through the three layers, outermost to innermost. The outermost is what kind of cells? Cytotrophoblasts. The second layer is the basement membrane, good, or basal lamina, same thing. And the last layer is the endothelial cells. Exactly. Perfect. Good job, guys. Okay, so all of this is actually only one layer of the placenta. Okay, all of this is the chorionic villi, and that's the innermost layer of the placenta. I'm going to show you in the next slide the second and third layer of the mature placenta, okay? Okay, so let's recap. We're over here right now. We are at a villus. These are just a bunch of villi over here, the chorionic villi. As you can see, they're tertiary villi because they have the fetal blood vessels right over here in the middle. And so basically what we zoomed in right now is we really zoomed in on this part. Remember I showed you that there's maternal blood right outside the cytotrophoblast. So imagine that this is the endothelial cells. Okay, and then imagine that these are the cytotrophoblasts, or sorry, the syncytiotrophoblasts over here on the outside. And then this part right over here is the maternal blood. Okay, and look, we actually see something that we've seen before. Look at the lacuna. Remember, I kept emphasizing to you, lacuna is maternal blood, lacuna is maternal blood. The reason that I was doing that is because look at what the lacuna is doing. The lacuna is draining into over here, and it's surrounding my chorionic villi. And that's why over here in my previous diagram, you see I have the maternal blood. This is maternal blood coming from the lacuna. Okay, so this is lacuna, it's draining inside, it's going past the syncytiotrophoblast, it's going past the basal membrane, and it eventually the gases or whatever are going to diffuse across into the umbilical endothelial cells. That's what we've just covered. That's where we're oriented right now, okay? The second thing that I want to talk about, that was the chorionic villi, that was that part right over here. The second part of the mature placental membrane that I want to talk about is this spot right over here, which is called the intervillus space, okay? The intervillus space is over here, and it's a little bit more outer compared to what we were talking about before. And all I need you to know about the intervillus space is that it's made up of maternal blood. All I need you to know about the intervillus space is that it's where the lacuna drains, and all of this over here is the maternal blood. What they're trying to show you specifically is that these lacuna over here that are orange is oxygenated blood. So the oxygen is going to diffuse across. It's going to enter the umbilical vein. Yes, I'm saying umbilical vein because the umbilical vein is oxygenated, okay? And then that umbilical vein is going to go to the baby for oxygen. The gray lacuna that they're showing you is actually filled with deoxygenated blood, and basically it's going to drain the deoxygenated blood from the fetus back into the lacuna. And once they're back into the lacuna are going to drain back into endometrial veins so it can go back to the mom, so the mom can oxygenate it, right? Because babies can't breathe. They can't oxygenate themselves. So again, the first layer was the chorionic villi. We went into a lot of detail about the chorionic villi. Second thing I need you to know is that surrounding the chorionic villi is this area right here, the intervillus space. Intervillus space is made up of maternal blood, both oxygenated and deoxygenated, okay? And then the third layer of the mature placenta, you already know, is the decidua basalis. This area right over here, you can see all the lacuna that we talked about over here. And you already know the decidua basalis is the maternal part, right? Is the part that used to be the endometrium. but now it's matured and it's decidualized. And all you need to know about it is that it hosts the lacuna. That's pretty much it. There is one more thing that I want to talk about. So again, just to cover, we went over chorionic villi, we went over intervillous space, we went over decidua basalis. These are the 3 layers of the mature placenta, okay? Next thing I want to talk about is that these villi just can't be sitting around loosely. They need to be anchored. And if you look over here, you'll notice that this villi, or this villus, is actually anchored to the decidua using something called a cytotrophoblastic shell. So all you need to know is that some of the villus, they're not going to just be sitting around loosely. They need to be anchored to the mother side, right? Otherwise, it would just break off. So the way that they connect is using an anchoring villus, and the anchoring villus is made up of a cytotrophoblastic shell. That's what I need you to know right over here. I'm going to quiz you on this in the next slide just to make sure everyone's keeping up. Okay, so this area right over here, what is this called? Or sorry, not this part, this part. What is this layer called? The second layer of the mature placenta. No, not this part. Sorry, that's what I was showing before. Okay, not the part with the veins, but like the part surrounding the umbilical vessels, like this. Intervillous space. Yeah, sorry if that wasn't really clear. Okay, what kind of blood is in the intervillous space? Maternal blood, good. And then for the deoxygenated blood, for example, how do they return back to the maternal circulation? Through what? It's represented by these gray things here. Yes, veins, endometrial veins specifically. Okay, what's the third layer of the mature placenta? Yeah, decidua basalis, good. Okay, and then part of the decidua basalis, or like, basically what's this called right over here? This villus has a specific name. What kind of name is it? Its role is to keep itself bound to the decidua. What do you call this? What kind of villus? Anchoring villus. And it's made out of what? Anchoring villus, yeah, and what's it made out of? Yeah, cytotrophoblastic shells specifically. Perfect. Okay, so for this, we talked about the syncytiotrophoblast. I told you to keep it in your brain because it's going to produce actually multiple hormones. I already told you about one hormone. What was that hormone that I asked you about, or that I told you about, is made by the syncytiotrophoblast before week 10 specifically? HCG, wonderful, guys. Really happy. Okay, so yes, you can see it over here. The first hormone, specifically before around 10 weeks, is HCG. It's made out of the syncytiotrophoblast, which is the outermost cells. Here's what I need you to know. This is really, really important, okay? HCG is structurally very, very similar to LH and TSH. Okay, I need you to remember that. I'm going to ask you about it later on. HCG is structurally similar. I believe it shares an alpha subunit to LH and TSH. And you can imagine if it's structurally similar, it actually has the capability of stimulating the receptors of LH and TSH. So specifically what that's going to look at is that HCG can stimulate the LH receptors of the Leydig cells or the adrenal cortex, or it can cause hyperthyroidism, from stimulating the TSH receptors. So I'm going to ask you about that later. HCG resembles LH and TSH, okay? Okay, and then how many weeks approximately does HCG last? Up until what week? 10, roughly. Sometimes it's like 8, but yeah, roughly. They also want you to know that HCG is responsible for the nausea in the morning. Okay. Okay, so after week 10, what does the syncytiotrophoblasts start to make? What hormone? Is this why pregnant women may develop hyperthyroidism? Perfect, exactly. This is the mechanism for that. Progesterone, exactly. After week 10 or around week 10, the syncytiotrophoblasts is going to start making progesterone, okay? And it's just responsible for maintaining the pregnant uterus. Okay, here's something that they want you to know. Because basically when you're pregnant, you want the progesterone to be able to be unlimited. You don't want there to be a limit for the amount of progesterone that you're able to make, okay? So because of that, the rate limiting protein is actually not present in placental steroidogenesis. In other words, the placenta can make as much progesterone as it wants. It's not limited by this protein over here called the star protein. I'm going to quiz you on that later on, okay? And this is just some of the roles of progesterone. It's required for implantation and breast growth and differentiation. Okay, so just to recap, when we're talking about the syncytiotrophoblasts or the placenta, can make as much progesterone as it wants, because it does not have what protein? Star. Perfect. Perfect. Yep. Here's what I think they want you to know. So over here, you can see syncytiotrophoblasts, which means that this is part of my placenta. Okay. And basically, I think what they want you to know is that the cholesterol to make the progesterone comes from the mother. Okay. So the mother's going to transfer cholesterol to the syncytiotropoblast, and the cholesterol is a precursor to progesterone, and the progesterone is going to go back to the maternal blood vessels. I think that's the main thing that they want you to know from this. Just the progesterone is made by syncytiotrophoblasts, and the cholesterol comes from the mom. Okay, just a very quick recap. I want you to know that, because I don't know if you guys have learned this, but we have our hypothalamus, which usually secretes CRH. CRH goes to the anterior pituitary gland. Anterior pituitary gland goes to the adrenal cortex, which is this gland over here, in order to make cortisol. So this is how in normal bodies like me and you, this is how we make cortisol. We have CRH from the hypothalamus, goes to the anterior pituitary to make ACTH. ACTH goes to the cortex. Cortex makes cortisol. And it's usually a negative feedback loop inside mine and your bodies. I'm not talking about a fetus right now. Second thing I want you to know is that cortisol is really, really important for lung maturation. Really, really important for lung maturation for the fetus, basically. And it's also important for GI, but it's more clinically relevant to know the lungs, in my opinion. Okay, here is what I think that they want you to take away. I know this slide looks really, really intimidating. Here's what I think they want you to know. I think they want you to know that the fetal pituitary gland is going to release ACTH. Remember, I showed you in the previous slide, it's the same thing that happens to our body. It's going to go to the adrenal cortex. Again, same thing that happens in our body. There's a precursor of progesterone, and eventually this progesterone is going to be made into cortisol. Okay, I think all they want you to know is that cortisol is made inside of the fetal adrenal cortex. Okay, and that progesterone is like a precursor to it, and it's stimulated by the fetal pituitary gland. Okay, now imagine this is happening on the fetal side. The mother side, because the mother's a normally fully developed human being, she's also having that on her side, right? She's having the ACTH going to the adrenal cortex in order to make the cortisol. I think the main thing that they want you to know about this is that the cortisol from the mom does not go to the baby, or at least it's very, very limited. And the cortisol from the baby does not go to the mom. And you can imagine the reason that this is. It's because cortisol is released in times of stress. So imagine the mom's really stressed out for something. You don't want the cortisol to go to the baby and have the baby get hyperglycemic, right? That's not going to be good. Same other way around. So basically what you should know is that the cortisol that's made from the mom stays in the mom. Cortisol that's made from the baby stays inside the baby, okay? And the release or the process of them being made is very similar to what's happening in our body. There is one small difference, but I'll go over that a little bit later. That's point one. I think point two that they want you to know is that cortisol, oh, sorry, I didn't mention that the reason that the cortisol does not pass the mother or the cortisol from the mother doesn't pass to the baby is because it needs to go through the syncytial-trophoblast, right? For fetus to the mom, it needs to go through the placenta. And basically, the placenta is going to stop the cortisol and convert it to cortisone. Okay, and this cortisone can't go anywhere. So that is how the placenta plays a role of making sure that the cortisol that comes from the baby stays inside the baby, and the cortisol that comes from the mom stays inside the mom. Okay, because it's going to convert cortisol to cortisone. Okay. Second thing I want you to know is that the placenta is not making its own cortisol. Notice that it doesn't have this protein over here, CYP17, which means that the progesterone over here cannot be made into cortisol. So I think what they want you to know is that the baby makes its own cortisol inside its own adrenal cortex. It can go to the placenta, but that's where it ends. It's not going to cross the placenta and go to the mom. That's what I think they want you to know. Syncytial-trophoblast, placenta cannot make cortisol. I'm going to contrast this to something later, so make sure you know that. Okay, now I want to move on. So we talked about cortisol. We talked about how ACTH is going to go to the adrenal gland. It's going to stimulate cortisol synthesis, right? I want to talk about another hormone that's also made by the adrenal gland, but it's made from a different layer of the adrenal gland, specifically the zona reticularis. This layer of the adrenal gland is going to make androgens or sex hormones like testosterone and sometimes even estrogen, or actually very importantly, estrogen. Okay, so again, we're still talking about the adrenal gland. It's just this time we're focusing on a different layer and we're focusing on a different hormone. This time we're talking about the androgens, okay? And what I want you to know for background knowledge, what I want you to know about for background knowledge is that once ACTH stimulates the adrenal gland, it's going to make this precursor called DHA, and DHA is a necessary precursor to estrone. Okay, DHA is a necessary precursor to estrone, which is sort of like estrogen. Okay, that's what I want you to know. It's a precursor to estrogen. It's really important to know. Okay, so we're back here. This is my placenta, and this is like the fetal side of things. Here's what I want you to notice. Okay, we're having the same thing happen. We're having fetal pituitary secreting ACTH. It's going to the adrenal gland, but it's stopping at DHEAS. That's what I want you to notice. The fetal adrenal cortex is going to stop at DHEAS. I need you to know this. The fetal adrenal cortex not going to make estrogen by itself. The fetal adrenal cortex not going to make estrogen by itself. This is the opposite of what was happening with cortisol. The fetal adrenal cortex was making its own cortisol. No, that's not what's happening here. The fetal adrenal cortex cannot make its own estrogen. Instead, it can only make up to DHEA, which is a precursor. The DHEA needs to go to the syncytial-trophoblast, which is the placenta, or the part of the placenta. It needs to have this enzyme, steroid sulfatase, which will then convert the DHEA to estrone, which is a form of estrogen. Okay? What am I trying to tell you over here? I'm trying to tell you over here that the syncytial-trophoblast, or the placenta, makes estrogen. If I ask you where is estrogen produced during pregnancy, you cannot tell me fetal adrenal cortex. You have to tell me placenta. Placenta makes estrogens. Okay? And then just something else, one small caveat, is that some of the DHEA actually comes from the fetal liver. So instead of the adrenal cortex, it's going to be the fetal liver, and it can make estriol, which is a different form of estrogen. So again, estrone and estradiol come technically from the precursor from the adrenal cortex. In contrast, estriol comes from the fetal liver. But most of the DHEA or most of the estrogens still come from the fetal adrenal cortex. So this is more high yield to know. Okay? And then, yeah, obviously these estrogens are going to go into the maternal circulation. They're going to help with breast development and all those things. Okay, I know that was a lot of information. So just going back over here, if we're talking about estrogen, I want you to pay attention to this enzyme over here, 3-beta-HSD. Second, sorry. Or actually, could you guys just tell me what is the precursor to the estrogen that I kept saying? What was that precursor? Good. DHEAs. Okay, yeah. And then I don't know if I mentioned this, but basically this part over here, CYP17, the syncytiotrophoblasts, they don't have that. So the precursor can only be made from the adrenal cortex. Okay, the precursor, the DHEA, can only be made from the adrenal cortex. And then it goes to the syncytiotrophoblasts, and then it becomes estrogen using steroid sulfatase. Okay, what stimulates DHEA from the anterior pituitary gland? What's the name of that hormone? Good, ACTH. Yes, and it's going to make, so the ACTH is going to make the precursor DHEA. Good. What's the name of the enzyme that converts the DHEA into the estrogen? There was one I wanted you to know. Steroid blank. Sulfatase, good. Yeah, it's the name of the enzyme is sulfatase. I'll show you over here. Over here, the DHEA, see, it needs to be processed by steroid sulfatase. Okay, and then after that, it forms what? After the DHEA, what's the end product of DHEA? Estradiol, good. And where's estriol from? Remember I told you this one's a little different. Good, by fetal liver. Okay, something else that I'm going to go over later in the next lecture, but I want you to be introduced, is that I want you to know that estrogen stimulates prolactin. I want you to know that there's a relationship between estrogen and prolactin. I'm going to go over it more in the next lecture, but I just want you to be aware of it. And these are just some of the functions of estrogen. It can uteroplacental blood flow, prostaglandin, oxytocin receptor sensitization. Okay, so we're going to go on to the next. Do we need to say estradiol or can we say estrogen instead? I think estrogen is enough at your level, but if you want to be specific, you can say estradiol because they're all forms of estrogen. I think it should be accepted. Okay, we're going to move on to the next hormone, which is called human placental lactogen. Okay, before we go on to human placental lactogen, let's just take a step back. Let's go back to HCG. You guys remember HCG? What were the two hormones that were very similar to HCG? LH is one, good. TSH, perfect. Yeah, I just wanted to revise that really quickly so that you guys know. Now forget HCG, let's go back to the human placental lactogen. Okay, the reason I was bringing that up is because basically, first let me tell you what human placental lactogen is. Human placental lactogen, from its name, you can tell it's kind of related to sugar, right? Lactose kind of means sugar, or it's a form of sugar. What I want you to know about human placental lactogen is its main goal in life is that it wants to keep the glucose levels in the mom very, very high. Okay, human placental lactogen, the main purpose of it is to keep the glucose levels in the maternal bloodstream very, very high. Why does it want to keep the glucose blood levels very, very high? So that the glucose can go to the baby, right? The more glucose in the bloodstream in the mom, the more glucose that's going to go to the baby. Okay, that's the main thing I want you to know about that. Okay, if you know that, then you can sort of rationalize in your brain, oh, of course, since it wants to keep the glucose levels so high in the mom, it's going to play a role where it's going to contribute to something called insulin resistance. Think about what that means logically. If I'm resistant to insulin, it means, or the mom is resistant to insulin, she can't lower her blood sugar because the role of insulin is to lower the blood sugar, right? And if she can't lower the blood sugar, exactly like someone said in the chat, she's going to get gestational diabetes. So human placental lactogen plays a very large role in gestational diabetes. That's very, very high yield to know. When gestational diabetes pops in your mind, I need you to think human placental lactogen. I need you to think human placental lactogen is trying to raise the glucose as high as it can go so it can go to the fetus. Listen to what I'm saying. I'm saying the glucose needs to go to the fetus, not to the mom. That's also really important to know. The glucose is elevated in the maternal bloodstream so that it can go to the fetus. Why am I pointing that out? Because the human placental lactogen is actually going to increase free fatty acids for the mom. The mom uses free fatty acids, the baby uses glucose. That is also a really important thing to know. So if you read the slide, everything should make sense. Lipolytic actions so that she can use free fatty acids for the mother. In contrast, it is going to inhibit maternal glucose uptake because it doesn't want the glucose to go to the mom. It wants the glucose to go to the baby. Okay? The other thing, the last thing that I want you to know about, just like HCG is similar to LH and TSH, HPL is similar to GH and prolactin. I don't think that this should be too confusing because prolactin, lactogen, they already sound kind of similar. It should be obvious that they look kind of similar. The second thing is that growth hormone, if you're ever confused, think about what's happening to the baby right now. It's kind of like puberty, right? It's kind of like the baby's going through puberty. It's developing so quickly. Similarly, growth hormone is really increased when teenagers are going through puberty. So they play a very similar role. So it should make sense to you that they're structurally similar. Growth hormone also increases the blood sugar levels by a lot. So that's what I want you to know. HPL is similar to GH and prolactin. Okay. I think this is close to the last slide. Okay, remember this is the fetal adrenal cortex. It makes DHEA. The DHEA has to go to the placenta, specifically the syncytiotrophoblasts, to make estradiol. The estradiol then goes to the maternal circulation to create what? Okay, I guess I didn't emphasize that a lot, but remember I talked about on the slide before, estrogen and prolactin play a very close relationship. Estrogen is going to stimulate prolactin release. I don't think I emphasized that enough. I'm going to emphasize that more going on. But make sure you know that estrogen or estradiol goes to the maternal circulation to increase prolactin, okay? Okay, you see how the CRH is over here in the placenta? I didn't lie to you. There is CRH that's made from the fetus and the mother, but I think they want you to know that CRH is not only made by the hypothalamus of the mom or the baby. I think they want you to know the CRH also comes from the syncytiotrophoblasts. So again, in normal people like me and you who aren't pregnant or we're not fetuses right now, we make our CRH from the hypothalamus. But in this case, it seems like the CRH actually comes from the placenta. And then the CRH goes to the fetal pituitary gland, and that stimulates ACTH. Okay, same thing over here in the maternal side. CRH goes to the maternal anterior pituitary gland, secretes ACTH. ACTH goes to the adrenal cortex to make cortisol. All I need you to know, placenta makes CRH. Oops. Okay, you guys already saw that, but what's another hormone made by placenta that plays a role in insulin resistance? HPL, good. For maternal too, yes. The CRH also goes to the maternal circulation and induces cortisol synthesis. And just remember that the maternal and fetal cortisol, they don't interact with each other. Just keep that in mind. Okay, HPL. Does anybody know the last hormone the placenta makes? So what about the hypothalamus of the mom? The hypothalamus, I think, is also creating CRH, but I think it's also being stimulated from the placental CRH. Both of them are occurring. But I guess in pregnancy, maybe most of it is coming from the placenta or something like that. Does that make sense? Yeah, the last hormone is progesterone. Good, from the placenta. Okay, the next thing, for the baby, what does it use for energy? I told you that there was a specific thing that it needs to use for energy. It's related to the HPL, glucose. Good. And it also uses amino acids, just by the way. And then I also talked about this before, but specifically HPL, it's going to have an anti-insulin effect. It's going to contribute to insulin resistance. The Thing is, you still are going to have hyperinsulinemia, aka elevated levels of insulin inside the mom. And you're probably thinking, Hania, like, what are you saying? There's elevated levels of insulin inside the mom. Wouldn't she be hypoglycemic? My answer is no, because she has gestational diabetes, for example. This is going to make her insulin resistant. Okay, so yes, there's going to be elevated levels of insulin. That is true, but it doesn't matter that there's elevated levels of insulin because the insulin can't do its job because there's insulin resistance because of the HPL. Okay, but there is going to be hyperinsulinemia. So it's like diabetes type 2, exactly, except this time it's from HPL. But yeah, you should know that there's high levels of insulin, and it makes sense why there's going to be high levels of insulin because your pancreas is still going to be noticing that there's very high levels of glucose. High levels of glucose is still going to trigger the high levels of insulin, right? There is still going to be beta cell hyperplasia. It still is going to be stimulated, in other words. Okay, last thing, what does the mom use for energy? Free fatty acid. Good, good. I'm glad that you guys got that. Okay, this is the last slide, I believe. Let's look at these graphs. Okay, let's see. This is week 10. Remember, week 10 was a really important thing. What hormone is increasing at week 10 and then is no longer needed after week 10? Could I go over prolactin? I can go over prolactin. It's going to be more in the next lecture, but all I need you to know for right now is that estrogen or estradiol stimulates prolactin. That's all I need you to know. I'm going to go over everything that prolactin does in the next lecture. Is that okay? Yes, you guys are getting it right, by the way. It's HCG. Okay, HCG. So now let's look at this. HCG stimulates the production of what? What hormone? Progesterone. So that's the second graph over here. Perfect. Yes. So what I want you to notice over here is that this level, this part over here, I know they didn't draw it. This level, the progesterone, would be from the corpus luteum. And then this really rapid increase would be from where? Where does the rest of the progesterone come from in pregnancy at around after week 10? Syncytiotrophoblasts. Good. Very precise answer. Good job. Okay. Here's a very similar hormone. It's also made from the syncytiotrophoblasts, okay? But it's only made when the syncytiotrophoblasts is mature. Does anybody want to take a guess of what this might be? Starts to increase. HPL, good job. And then this is the last thing. I don't think this is super high yield to know. I think the only thing I would know is that estradiol is the most abundant form of estrogen. I think that's the only thing that I would take away from this, honestly. Remember, estradiol comes from the adrenal cortex, and then, well, the precursor from the adrenal cortex, and then estriol comes from the liver. Okay. Sorry that I took kind of long in that lecture. We have a second one to go. Any questions about this lecture? Anything you want me to repeat? All clear. Okay. Do you guys want to break, or do you want me to just go on to the next one? You guys want to break? Okay. How long? Like 10 minutes? 5 minutes?